Hello all. Thank you all very much for coming, and it is really a great pleasure to be here with you and tell you a little bit about some of our work into pancreatic cancer. But before I start, I just thought perhaps not everybody knows what pancreas actually is and what it does. Just because it is kind of neatly hidden here, you can see in this pictorial it's kind of next to the liver, gallbladder, spleen, and hiding kind of under the spleen. But it does actually play two main roles. So the first role is in helping digestion. So the pancreas secretes a clear watery substance that contains several different enzymes. And so what these enzymes do is helps break down proteins, carbohydrates, and fats into fatty acids and cholesterol. And what it also does, it contains specialized cells that can secrete different hormones. And these hormones are critical in maintaining our blood sugar levels. And as you'll hear later on in the next talk, maintaining blood sugar levels is really important because we need a constant supply of sugar to our organs, provide energy so we can function, but at the same time, too much sugar can induce toxicity and cause toxicity in kidneys and other organs. So hopefully you can all agree that maybe it's not so anatomically obvious what pancreas does, but it plays a very important role in maintaining our health. Now, this has also been touched upon a little bit earlier, but one of the sort of downsides of the increased average life expectancy. So this is really with all the major discoveries that have been happening in the treatment of other conditions and other health-related issues. Unfortunately, simultaneously, both pancreatic cancer incidence and mortality rapidly rises with advancing age. And just to give you some of the facts, this is a disease with very dismal prognosis. So only 5% of patients survive, 5-year survival of these patients is only 8%. It is currently the 4th most common cause of cancer deaths. By 2030, it's predicted to become the 2nd leading cause of all cancer deaths. And that's mainly due to being mostly diagnosed at a very advanced stage. And so most of the patients actually die within the first year of diagnosis. And the current treatment options, which I'll mention a bit later on, only improve survival by a few months. And so that's really why my group focuses on trying to develop new treatment approaches and more tailored treatment approaches for this disease. And here's yet another statistic, but what this highlights is really that in comparison with some of the other cancer types, and so these arrows here just really show a shift or improvement in the overall survival rates that we're now seeing with these cancers. Pancreatic cancer is really lagging very much behind. So it's really only increased by 5% in the last few decades. So we have a really long way to go. And one of the issues with developing new treatments or finding really effective treatments for this disease is that this disease is molecularly very diverse. And so you don't need to know all of the details here, but what you can see is, so this is a cell, so a pictorial of a cell. So this is a cell membrane. Inside we have a nucleus. And what this really shows you is that each cell in our body has very complex molecular mechanisms that can determine what this cell will do, how it will move, how it will grow, and whether it will die. And so what happens in cancer is that all of these processes, or one of these processes, can become aberrant, and this can be caused by genetic mutations, for example, and then these things can then lead to cancer. And so we've actually done some of the work in this area previously, so we have performed, and I'll talk a little bit about it, but what we have found is that using whole genome sequencing analysis of patient tumors, we actually found that each patient tumor can be vastly different from another patient's tumor. And so the one-size-fit-all approach that we're currently using with treatments is just not the right way to go. And that's really what we work on now. So it's the development of more personalized treatment approaches for the disease. But I'll just really highlight this study. So it's been published a while ago, but it was a major finding because it's really driven sort of the approaches that we've now taken in trying to develop new therapies. And this is just showing you, so these are circular plots or circos plots. They're representing genome sequencing data. So here we have, so we've done over 100 patients as part of that study. But what does it all mean? So you have chromosomes actually represented in a circular manner, hence its circos plot. And each of these lines here show detected changes in this particular tumor DNA. So these could be pieces missing, so deletions of different genes inside this tumor. You can have chromosomes or genes that are aberrantly now jointed together. So these are these translocations, these huge lines moving from one chromosome to another. We can have amplifications, which means that chromosomes may be present in too few or too many copies. But what became clear to us when we sequenced this large cohort of patient tumors was that we could start to subclassify pancreatic cancer into at least 4 different molecular subtypes, just based on this sort of genome sequencing data. And this particular subtype we called an unstable tumor genome because it had over 200 different genomic aberrations present inside individual tumor. And just to show you, so just based on the evidence from some of the other cancers, we knew that these sort of tumors could be, or we hypothesized that in pancreatic cancer, therefore, they should also be fast growing. They don't seem to respond to one of the standard of care therapies called gemcitabine, but they may respond preferentially to specific DNA damaging drugs, things like platinum agents. And so just to show you that not all tumors actually look like that, so this is an example of a stable tumor genome that we've also sequenced as part of the cohort. So you can see very few genomic changes happening in this particular tumor. And this is really the pipeline that we used as part of this project to guide therapy for patients. So it involved taking patient tumors, performing, in this case, DNA sequencing. We're now also performing other types of analyses, so analyzing the RNA, analyzing the protein in these tumors, detecting mutations, molecular alterations, and then trying to choose the right, in this case, chemotherapy drug to tailor to treatment for the patient. There's also an intermediate step here before you can do that. So you also need to validate or test your hypotheses in preclinical models. And so as part of this study, what we've also done is we've taken different patient tumors and we've established them in preclinical models, established preclinical models from them. So we have 80 different patient tumors either growing in culture. So this is 2-dimensional culture on plastic, and I'll show you some images of those later, or growing in mice, so that any sort of optimal or effective therapy that we find to work in our cultures, we can then test it in the mouse models before giving to the patient. Because you have to prove that your therapy really is effective in a whole body sort of system, organism. And so we were able to do this now for a few patients as part of that study, particularly using these DNA damaging drugs. So here we have the example. So this was a patient tumor that we've sequenced. It had an unstable genome. We created the hypothesis. So this was trying to predict response to treatment. We generated the model, so the patient-derived tumor model from this particular patient tumor. And so we both screened it. So this is the culture experiment, and then this is the mouse experiment. So just to show you briefly in the culture experiment, the unstable tumor culture, it shows you on the vertical axis, we have the percentage of cells alive post-treatment with different drugs. In blue, we used one of the standard of care therapies, gemcitabine. So that's just cut off at the end. Sorry about that. But you can see that the unstable tumor actually doesn't respond to this therapy. So most of the cells are very much still alive. However, it was very sensitive to the drug cisplatin, shown in red here. So that's actually marked here for the experiment that we've performed by growing these tumors in mice. So here we have the tumor volume on the y-axis, and you can see that in the presence of cisplatin, but not gemcitabine, these tumors just shrunk. So we were also very fortunate to be able to apply this strategy into the clinic. So unfortunately, this patient relapsed following the surgery. So this is the standard clinical path that was taken. the surgery, the gemcitabine therapy, chemotherapy, the patient recurred. So we then spoke to the oncologist who treated this patient, and he was happy based on our data and also based on the genomic sequencing data to then administer cisplatin as a targeted or tailored treatment approach. And this patient is still alive. Actually, this has not been updated, so this is now 4.5, almost 5 years post-diagnosis. So that was just showing on the CT image, there was a recurrence here, and then that recurrence completely disappeared following treatment with the tailored approach. And so what we're doing now is trying to work on the different types of omic data that we have in the laboratory that we've generated from these tumors and try to define new personalized treatment strategies, essentially for distinct either subtypes or even in an individualized setting, so per patient. And one of the drugs, again, it's not all about novel therapies. We have to learn how to use perhaps therapies that are successfully used in the treatment of other cancers. And so this was another study that we published reasonably recently, last year. So this is working on a drug called palbociclib, or PD0332991. I can't seem to be in control. I'm not in control of the anything really. Doesn't matter. So this particular drug targets the pathway which is called CDK4/6, or cyclin-dependent kinase 4/6 pathway. And so what we did here is we took a patient-derived cell line, so this is a cell line from the patient tumor. We then introduced a specific biosensor, and you can think of this as a traffic light. So these biosensors are really quite neat tags that we can insert into the cancer cells. The science behind this is really amazing. I mean, the disease is not, is a very hard disease to work on, but the science behind it is really very exciting. Anyway, so you can think of this as a traffic light. So basically the green cells mean that these cells are growing and rapidly dividing. Red cells that have stopped or have arrested. And then yellow is yellow, right? Get ready to go. And so what you can see below, if I can just start the videos. So this is the control setting. So it's as you expect. So tumor cells, we have a range of green cells, red cells, and they're just kind of rapidly moving from one phase to another. But what was really interesting is that when you treat with palbociclib, over a 16 to 24-hour period, so this is how long we filmed these cells for, they all shift to the red state, so they stop growing. So this drug was quite effective, at least in our culture models, to firstly block growth of these tumor cells in vitro, so in cultures. But we also know that pancreatic cancer, unfortunately, it's not just composed of cancer cells. It also contains quite a complex microenvironment. So that's shown here. So these are cancer cells kind of in purple, but then we actually have these so-called normal cells, but they're not normal. They've been corrupted essentially by the tumor cells to signal and to keep promoting tumor growth and tumor cell survival and also resistance. And so one of these, so what this microenvironment contains, it involves a matrix, so fibrotic sort of matrix that involves collagen, for example. It contains immune cells and some of the other stromal cell types, vasculature. So these tumors really create their own supply. It's almost like an organ, corrupted organ, and then they take off and grow. So one of the functions, though, of this fibrotic matrix is that it can limit drug penetration. So when you're trying to treat with a specific therapy, this can then really limit how effectively your drug can actually enter the cancer cells and kill them. And so this is now showing you just some tumor sections of the tumors that we have treated in mice with these drugs. So with palbociclib, shown here. So you can again see even in vivo, so in the mouse model, we've been able to effectively block the growth, also kill some cells with this drug. But what's really interesting is that at the same time, so purple is showing this fibrotic matrix. So we've been able to mark the fibrotic matrix using complex microscopy. You can see that following treatment with palbociclib, this is now dramatically reduced. And then when you follow up these tumors and you look at survival of these models, so you look at how effective your drug is working in these different patient-derived tumor models, we can see that in the top, especially when guided by a specific biomarker. So this protein is called RB. We're looking for presence of this biomarker in the tumor sections just by protein analysis. This is a critical component, so RB is a critical component of the CDK4/6 pathway that palbociclib targets. So hence, that's kind of how we identified it. But what's really interesting is that you can see that either palbociclib alone, shown in red, or the combination with chemotherapy, gemcitabine, which is really what we were trying to combine it with in terms of its clinical implications, both of them effectively inhibit tumor growth in two different volumes, in two different models that carry this biomarker. What's also really important is that in the negative tumors, so tumors that are negative for this protein, they've lost the expression of this protein, this drug combination doesn't work. So what we're saying is, again, this is going to be a very tailored treatment approach, guided by specific biomarkers. This is certainly not for all comers. And I just thought I'd leave you with this. So this really just highlights sort of the work that we're trying to do, really. So we're very interested in investigating novel therapies. So these are therapies that are potentially in preclinical pipelines to try and see if we can treat specific pancreatic cancer subtypes and actually get meaningful improvements in outcome. We're also very interested in repurposing some quite random, perhaps kind of out of the box therapies that are used in the treatment of other conditions. So for example, antifungal drugs, one of them in particular, itraconazole, is we're very interested in. We're looking at molecular mechanisms behind the chemotherapy resistance, also resistance behind other therapies. So we know that pancreatic cancer does not respond to immunotherapy. We're trying to figure out why, because when immunotherapy works, then it really leads to long-term improvements. So if we can sensitize pancreatic tumors to immunotherapy, then that may actually be a massive bonus in terms of improving actual patient lives. I'll leave it there. Thank you very much.